Hello friends, in this video lecture I will talk about the Shine-Dalgarno sequence. It is in short called ST sequence. So ST sequence is a ribosomal binding site that helps in translation initiation process in prokaryotes. So the ST sequence it helps in translation initiation. It helps in translation initiation in prokaryotes. So the term translation, that is the synthesis of proteins, and it is the second step of gene expression. We know that when gene is expressed, a particular protein or polypeptide is synthesized. So it is the second step of gene expression. In the first step, that is the transcription. In transcription, mRNA is synthesized. In the process of transcription, from DNA template, mRNA is synthesized. So this is the DNA molecule, and in the process of transcription, we get the mRNA molecule. And then this mRNA carries the genetic information from the DNA, and this genetic information is converted into or translated into the sequence of amino acids. Then the second step, the information carried by mRNA is translated into the sequence of amino acids that forms the polypeptide and protein, polypeptide. So this process is called translation. So the first requirement that the mRNA, for protein to be synthesized, we require mRNA. Then mRNA is translated into the proteins. So in the translation initiation process, the ST sequence helps in the translation initiation process. The ST sequence, so let me write the sequence that is found in mRNA of prokaryotes. Suppose this is a prokaryotic mRNA, and the ST sequence is a highly conserved consensus sequence. It is a highly conserved consensus sequence, right? Found in the prokaryotic mRNA, and it is located 8 base pair. It is located 8 base pair upstream of the start codon, that is AUG. Suppose this is the codon AUG, and this is the 5' end, and this is 3' end, and the ST sequence is found in 8 base pair upstream. This region, if the gene starts from here, this is the +1, then this is the upstream region. In this direction is found the ST sequence, means in the upstream region. This region also called UTR, that is the untranslated region. So the ST sequence is found here, and let me to write the sequence. That the ST sequence is AGGU. This is the 5' and this is 3'. So this sequence is found in this upstream region, right? So how it helps the mRNA to bind on the ribosome? Then we will talk about the structure, the composition of ribosome. So we know the ribosome is made up of 2 subunits. One is the larger subunit and the other is the smaller subunit. The prokaryotic ribosome, that is the 70S type, and its smaller subunit is 30S. Then this subunit of ribosome means the ribosome is composed of 2 substances, 2 components, that is the ribosomal RNA and proteins. These are the 2 major components of the ribosome. So in 30th subunit of the ribosome, what type of ribosomal RNA is found? So the ribosomal RNA found in 30th subunit of ribosome is 16S ribosomal RNA, right? So this is single stranded, and the mRNA is also single stranded. So this sequence, the ST sequence, must bind with a particular site of the 16S ribosomal RNA. Means this ST sequence is complementary to the sequence of 16S ribosomal RNA. So that sequence, that is called ASD sequence. ASD sequence. And ASD sequence is found in 16S ribosomal RNA. So the ST sequence and ASD sequence are complementary. So this sequence AGG-U easily binds with the ASD region. The ASD sequence is found in 16S ribosomal RNA, so that the mRNA easily attached to the ribosome. If ASD sequence is AGG-U, then the ASD sequence is 3' UCC A 5'. So in this way, the ST sequence of prokaryotic mRNA form complementarity with the ASD sequence of 16S ribosomal RNA, thereby it easily binds to the smaller subunit of the ribosome. So this is the prokaryotic initiation process, which is helped by, and that is the process is facilitated by the ST sequence. So what is ST? So this ST sequence is named after the scientist. Two Australian scientists named as John Shine and Dalgarno. These two Australian scientists discovered this SD sequence, and in this way, the protein initiation, the initiation process of translation that begins in prokaryotic cell. So, what about the eukaryotic cell? How this event is carried out in eukaryotic cell? So now, let's have a discussion on eukaryotic initiation process, or how the mRNA binds to the smaller subunit of eukaryotic ribosome. So the eukaryotic organism, the eukaryotic cell uses a different mechanism for binding of mRNA to the smaller subunit of ribosome. So this sequence that involves the Kozak sequence. So in eukaryote, we get the Kozak sequence. The mRNA contains the Kozak sequence. So it is also named after the name of the scientist. So it is discovered by one American scientist, Marilyn Kozak. Discovered the Kozak sequence that is found in the eukaryotic mRNA. Suppose this is the eukaryotic mRNA, this is the 5' end, and this is 3' end, and this Kozak sequence is located towards the 5' end of the mRNA. So let me write the Kozak sequence, that is GCC 5', GCC ACC AUG, then G 3'. So this is Kozak sequence. So this sequence is present towards the 5' end of the mRNA. The Kozak sequence, Kozak sequence, it surrounds the initiation codon. So in this sequence we get the initiation codon, that is AUG. The protein synthesis starts from here. It surrounds the initiation codon. Okay, right? In case of eukaryote, the ribosome scans this mRNA from the 5' end until it encounters the start codon. The ribosome scans from this side until it encounters the AUG codon, and this AUG codon is inside the Kozak sequence. So the Kozak sequence helps in this process. And another protein factor, that is the initiation protein factor, that is EIA4E, that interacts with EIA4E, this initiation protein factor. The protein factor interacts with the Kozak sequence, So this protein factor also helps in binding of the mRNA to the smaller subunit, basically the 40S subunit of the ribosome. And this EIF4E also binds to the 5 prime end. 5 prime end means in the eukaryotic mRNA. So in eukaryotic mRNA, that is a post-transcriptional processing is required. We know that after synthesis of mRNA, a 5 prime cap at the 5 prime end, a 7-methyl guanosine cap, 7-methyl guanosine cap is added to the 5 prime end. So this EIF4E also binds to this cap region. So what is the role of this cap? So the capping helps the ribosome to recognize and binds the mRNA. So it helps the ribosome so that the ribosome can recognize the mRNA and binds to it. So the role of in eukaryote, there is the role of 5 prime capping and this protein initiation factor EIF4E. They help in this binding process, the binding of this mRNA to the smaller subunit of the ribosome. So in this way, this binding occurs in eukaryotic cells. Now we will take a comparison between the ST sequence and Kozak sequence. So now we will see the comparison. We will take a comparison between the ST sequence and Kozak sequence, right? So the first point, that is the ST sequence, it is a ribosomal binding site found in, it is a ribosomal binding site found in prokaryotic mRNA, and it is found in, this Kozak sequence is found in eukaryotic mRNA, helps in the translation initiation process. Then its sequence is the SD sequence, that we knew that the SD sequence is AGGU 5 prime and 3 prime. This is the SD sequence, and the Kozak sequence is GCC ACC AUGU. This is 5 prime, this is 3 prime end, and this is the initiation codon. So it is located 8 base pair upstream. The ST sequence is located 8 base pair upstream of the start codon, and it is located towards the 5 prime end. It is located towards the 5 prime end of the mRNA, and it is discovered by 2 Australian scientists, John Shine and Dalgarno, and this was discovered by American researchers Marilyn Kozak. And we can give the example that so ST sequence binds to the 30th subunit of the ribosome, and this Kozak sequence binds to the 30th subunit of the ribosome. And we can give example as it is found in prokaryotes. So chloroplast, mitochondria, bacteria, archaea. So in chloroplast and mitochondria, these are found in eukaryotic cells, but the ribosome is 70s. So the smaller subunit is 30s. Means in the chloroplast and mitochondrial mRNA has ST sequence, and in this Kozak sequence we can take example as it is found in eukaryotic organisms. So in this way, the ST sequence in prokaryote and the Kozak sequence in eukaryote, the ST sequence in prokaryote and Kozak sequence in eukaryotes helps in the initiation process, in the initiation of translation process, means specifically helps the mRNA to bind to the smaller subunit of the ribosome. So I hope this video will make you understand the topic. Thanks.